My name is Paula Nerlich. I'm a member of the Hub for Biotechnology in the Built Environment and co-founder of our BioFutures group. And I would like to start with a question. So what if we could design meaningful interactions between humans and the non-human within the everyday space of the built environment, focusing on mutualistic symbiotic modes of being? Today, I'm presenting the collaborative design research project, Human Bacteria Interfaces, Designing for Microbe-Human Interaction in the More Than Human Coexistence. Our project is funded by Driving the Human, and together with fellow researchers from the Hub for Biotechnology in the built environment, our project explores pathways to design for microbe-human interaction in more than human coexistence. So introducing Ali. Co-created by Anne-Sophie Belling, Bea Delgado Corrales, Romy Kaiser, and me, Paula Nerlich. Ali is the first prototype created as part of a two-stage development process towards a human-bacteria interface within the course of 1.5 years. The human-bacteria interfaces, short HBI, concept examines how multimodal interactions between humans and microbes can elicit novel ways for humans to meaningfully collaborate and coexist with a non-human within the built environment. Specifically, HBIs are tangible living interfaces consisting of microbial consortia that interact or respond to stimuli from the surroundings by emitting signals accessible to us humans through touch, smell, and sight. And with our HBI, we explore designing through and with the other, encountering more than human realities that invite us to dive into new perspectives and to understand the other's needs, perception, and possibilities in interaction and cohabitation. By aiming for multi-species and more than human perspective, a new understanding of the other is nurtured, might it be human, non-human, living, non-living, or whole ecosystems. And this allows to challenge current eco-social and political systems that not only affect existence on Earth, but also humankind's journey beyond, and feeds new thinking into modes of multi-species cohabitation and negotiation between diverse agents. In our HBI, the main non-human agents are microbes. And within our narrative, we aim to decenter human agents and allow for a disassembly of established narratives of bacteria and microbes as inherently bad or harmful in the existence of highly complex, decentered ecosystems. Instead, we emphasize human nature as a dynamic mode of becoming, always changing and sustained by its interaction and coexistence with microbial beings, externally and internally. And we seek to support a shift in our perceived relations with the other than human, a shift that foregrounds humankind's vital reliance on the more than human to survive, thrive, and carve out more equal and caring and restorative futures. So at the heart of our human-bacteria interface concept is the idea of examining potential new relationships with microbes that are facilitated by symbiotic ways of living together. And through an interdisciplinary approach, including material design, creative technology, and synthetic microbiology, we utilize design and technology interventions as meaningful ways to explore relationships with a more than human world. And this challenges design and technology interventions that aim to facilitate a world of self-sufficiency of humans from the more than human world, and instead emphasizes our interdependence and interconnectedness within a complex system of nature. Further, working with living organisms challenges to constantly reflect upon how we design with the living and related duties of care, accountability, and responsibility. So what if living interfaces consisting of microbial consortia could create an ambient living intelligence that responds to its human inhabitants and surroundings? Now, when Ali, our artificial living intelligence, detects stimuli in its surroundings, it will respond by activating fluorescent proteins, resulting in an atmospheric glow, making an otherwise invisible interaction visible to the human cohabitant. Our Ali prototype was defined through a conversation with the ways in which microbial organisms sensorially and habitually engage with their surroundings. And by making such elements a central part of the initial phase of ideation, potential non-human narratives were defined and made part of the design and knowledge process. And ways of living with the non-humans were therefore examined through an appreciation of the other's alterity. So our HBIs are imagined to encompass a continuum of diverse potential implementations within habitats. However, the HBI prototype presented today, our Ali, specifically focuses on using SCOBY, a symbiotic culture of bacteria and yeast as a medium for HBIs to facilitate human microbial interaction through external stimuli and light. And this microbial mix is mostly embedded in a cellulose structure of bacterial origin, forming a jelly-like matrix, which provides an interface for microbial conversation and communication and interaction. And you see the SCOBY matrix just there. Some people might know from kombucha brewing, quite yummy actually. So Ali's process of creation goes through different stages in the lab and outside of the lab. And as I mentioned, we are at the Hub for Biotechnology in the built environment. So we have the fantastic opportunity to, even as a textile designer like me, dive into the microbiology process at our Hub labs, which is just amazing. So in the next steps, we would like Ali to react to a range of molecules and stimuli. And this includes a series of molecules that can be pollutants or stressors or relevant particles for other reasons. They might also include other types of stimuli. So that could be temperature, like heat or a low and high pH. And as a proof of concept of this interaction, we will design a plasmid, an extranuclear DNA fragment that contains an enzyme that produces light or color. And once reacting to a simple stimuli like sugar presence, this enzyme could be a fluorescent protein of the GFP family, which has different colors available that you can also see on our slide here. And it might also be any other chromophore, really. So that color will be present if the enzyme is reacting to the stimuli. And this way, the bacteria, or we will imagine the bacteria to light up if the stimuli is present, giving us that information that we could not otherwise detect through our human bodily senses. In the next step, the selected bacteria that produces bacterial cellulose will be grown in vitro, whilst an E. coli strain will be transformed with a plasmid. And once these cultures are optimized, the next step will be co-culturing them so that the visual effect is integrated in the bacterial cellulose. And the degree of fluorescence and color and co-culture ratio will be optimized. We'll also look at different color combinations. So imagine, what happens if you merge yellow and blue. So we get potential of 3 different signals. Yeah, this is a trial and error approach until we're really finding, hopefully, fingers crossed, the right combination of microorganisms that thrive and detect stimuli and produce signals readable by us. And after developing the GMO, the microbial consortia will be grown in an organic way, forming a bacterial cellulose matrix that will light up or change color after being exposed to the inducer. So for the final stage, we will optimize the growth and resistance of Ali as well we also look how to facilitate Ali in the final prototype. So this is really aimed to be a proof of concept, which means that at later stage, our research could lead to its application within the environment, and also that it can detect the environmental stimuli. Yeah, so it's a work in progress. With the cellulose matrix, it will serve as a receptacle for the interaction of Ali with its surroundings, as well as a diffuser matrix where the stimuli particles and molecules can access the microorganisms. So yeah, and the development will depend on the experiments done in the lab and how the DMO reacts to the stimuli once they are engineered or designed. But also we're looking at the legislations about GMO in the EU. Yeah, someone just laughed, so yeah. But we need to keep that conversation running, you know, and so it's a good one. Right, so next steps. Our design research team has begun the second stage of the development process, in which we currently refine the scientific process to design the HBI with bacteriocellulose. One of our expert advisors is on site. Thank you, Josh, for being here. And we are also co-developing a life support system in form of a unique bioreactor for our HBI for Ali. And further, we keep challenging ourselves, as I mentioned before, but I think this just needs to really come back into the conversations again and again. We need to challenge ourselves as interdisciplinary teams to really reflect about how we design with the living continuously. So I have to say, unfortunately, we cannot disclose many insights into stage 2 at this point due to an NDA, but we invite you to follow our journey and hopefully welcome you in Berlin in November 2022 to see our final work. And now I will share our concept video with you, if it wants to play.
What if we could design meaningful interactions between humans and the non-human within the everyday space of the built environment, focusing on symbiotic modes of being? What if such symbiotic modes of being could materialize through living interfaces that act as an ambient living intelligence, responding to its human inhabitants and surroundings? Introducing the human-bacteria interfaces concept and the ambient living interface, Ali. Human-bacteria interfaces are tangible living interfaces consisting of microbial consortia that interact or respond to stimuli from their surroundings by emitting signals accessible to humans through touch, smell, and sight. The Ali interface uses SCOBY, a symbiotic culture of bacteria and yeast, as a medium to facilitate human microbial interaction through external stimuli. The bacteria embedded in the SCOBY will be biotechnologically designed to translate stimuli into signals that are accessible to humans. When Ali detects stimuli in its surroundings, it will respond by activating fluorescent proteins, resulting in an atmospheric glow, making an otherwise invisible interaction visible to the human inhabitant. Embedded in the cellulose structure of bacterial origin, the SCOBY microbial mix forms a jelly-like texture, which provides a large-scale human-bacteria interface for the built environment. The bacteria embedded in the SCOBY matrix can be designed with biotechnology to react to a variety of molecules, such as pollutants, stressors, and stimuli such as heat, allowing for an array of implementations within the context of the built environment. Through the Ali interface, novel symbiotic relations between humans and microbial life are explored. Instead of perpetuating antimicrobial narratives, human nature is examined through its vital entanglements with the non-human microbial world, together transfiguring the metabolic landscapes of the built environment, as futures of eco-social renewal are examined through encounters with the microbial sphere. Join us in our vision of microbial futures.
Yeah, thank you very much. So yeah, hashtag microbial futures. I think I've got 2 minutes to take questions. Sorry, I must have run over. But you can also contact me via the email there, and you also find me via my website or our hub website. Yeah, are there any questions?
I'm wondering why you chose SCOBY. I mean, it's a very complicated organism to start off with, I guess.
Yeah, so,
oh, sorry.
Yes. So yeah, thank you for that question. So initially we started off with SCOBY because we are looking at a form of matrix that could be our interface, and whilst being sort of hosting and being co-created by these different organisms within. And also SCOBY, like as I mentioned, we know it from kombucha brewing. I also do that at home, and it is quite a stable environment. So for us to start off with, that seemed to be a good pathway to take. And yeah, so at the moment we're looking more into specific bacteriocellulose producing strains, but yeah, can't share too much about that yet, but stay tuned. Any other questions? Oh, yeah.
Thanks, Paula. I mean, I know your work a little from already being at the HBBE, but I think it's the first time I've seen you present. So it was really fascinating to hear it all. So thank you for that. I have a lot of questions actually, but the one that I will maybe ask for the moment is, you mentioned symbiotic relationships between humans and more than humans. And you are obviously having to engineer organisms as part of this process as well. And so I think there are a lot of complexities in what you're doing. And so I suppose what I'm asking is that perhaps maybe one of the really interesting outputs of this work might also be the process itself and how you go about thinking through the relationship that you are having with all of these organisms whilst you're working with them. I wonder if that's something that you've been considering.
Yes, absolutely. So first of all, as an interdisciplinary team with our creative technologist, Anne-Sophie, who will be speaking later, and then material and textile designers and our microbiologists. For us, starting this project, it was the first time we came together as a team, and then taking that journey is, I guess is for us almost more important than the final outcome. And looking at the language that we use, the processes that we go through in ideation, and you know, producing different forms of work, and co-creating with the organisms and so on. So that's definitely part of that. And I just forgot the second part of your question. If you could just, sorry.
I suppose really I was thinking that an interesting outcome for the project would be to somehow document the relationship that you are finding that you are going through with all of the complexity of the different types of organism, in a sense, in that you're having to, you mentioned this symbiotic relationship, and yet you are working directly with them and changing them in different ways. So it was about maybe how you will capture that part of the process.
Thank you for repeating that for me. So yeah, definitely, it's a in a way, a very scientific approach where we document every little detail. And for me, it's about learning how to work with these microbes. And I think it's also nicely connected to other presentations we had, looking at their perception of the environment, the requirements they have. And whilst we're trying to find a connection with them, there's also the question that was raised yesterday, what if the other doesn't want to? So it is a journey, and we're collecting the data, and hopefully, yeah, the outcome will be interesting, either supporting our vision, or we might learn actually the other's quite fine on its own. So yeah, I think I have to end here, but thank you so much. So for this session, we are inviting two microbiologists from the Hub for Biotechnology to speak about the research. So we have our new member, Chris Miller, speaking, sharing about his research into hydrophobic modification of bacterial cellulose. And after that, we've got Paul James sharing about diverse research projects, but one of them being about Perchlorate, which is a reactive chemical you find in Mars soil. So we're really looking forward to that one. So unfortunately, Chris could not join us today, but I'm going to share his video. And if you have any questions, please share them in the chat and we will forward them to Chris later on. And for Paul, he's here in person. So we look forward to a lovely discussion round. So let's get started. So the first on is Chris Miller.
Hello, my name is Christopher Miller, and this is my presentation on the in-situ modification of bacterial cellulose. In this research, I will cover the background of the research, my aims and objectives, the methodology, my results, and discuss them, and my conclusions, and any future works that I have planned. Bacterial cellulose, also known as microbial cellulose, is a biodegradable biopolymer. It differs from natural cellulose from plants in the fact that it is a pure glucose chain rather than having impurities such as lignin and hemicellulose. It's got an impressive tensile strength for a biopolymer, and the crystalline structure allows it to have a very tight hydrogen bonding network, which gives it its incredible tensile properties. As you can see on the side, there is a detailed map of a SEM image of the cellulose fibers and the glucose structure with the hydrogen bonding of which it is formed. So the uses and applications of bacterial cellulose are wide and varied, from medical dressings and bone regeneration, textiles and clothing in a design area, concrete additives and doping agents for building materials in place of synthetic polymers, bioelectric components when combined with metal ions, and even optical applications such as for contact lenses due to its optical properties. It's produced in a very green and environmentally friendly method, which is useful in manufacturing and industry due to the dangerous and oftentimes environmentally damaging chemicals used in normal production of polymers with similar properties. So the applications of space habitation are quite numerous. The ease of production means a reduced weight for transportation, as it only requires a feedstock to grow and the bacterium. The material can be grown on site and adapted to suit the needs of the material at the time, just by changing the modifications, which is what we're currently working on. The multiple uses in construction medicine mean you can produce multiple materials with differing downstream uses from essentially the same material, which also reduces the weight and amount needed for transport. The low waste is also very appealing, as it can be completely upscaled and recycled into other forms of the material for different areas. So you could basically have some wound dressings and then upscale and recycle them by breaking it down with cellulase into a, say, concrete additive. It can also be grown on biological waste, which is very appealing, as it means it's not only a complete circular economy, but it adds on top of and can use waste from other sources to produce more viable material. So there are two main methods for modifying bacterial cellulose. There is in-situ modification and ex-situ modification, shown by A and B. In-situ modification, the additive or modification is usually added during fermentation of the glucose molecules in the media, or it is a modification that is produced by the bacteria, or maybe even a cold culture method using another microorganism, which is the case with kombucha. And the ex-situ modification, where the bacterial cellulose is first produced and then chemically or mechanically modified to produce the modified form of cellulose. I've been focusing on the hydrophobicity modification so far, with cellulose acetylation being particular focus, as it can produce fibers with contact angles of up to 100 degrees, even 113, which is over the 90 degrees contact angle required for a material to be considered hydrophobic. And the selenization of cellulose, which involves using silicon molecules and silicon nanoparticles to coat or otherwise bind with the cellulose to produce a layer or incorporate it into the deeper pores of the material. So the aims and objectives of my research, I wish to produce a hydrophobic bacterial cellulose using the in-situ method of synthesis. Currently, I've been trialing different molecules, so 2 selenization routes and 1 acetic acid route. You can see the structures of the modifications in the figure. And these modifications are tetramethyl orthosilicate, tetraethyl orthosilicate, and acetic acid, said before. So the deeper methodology, there's kind of 2 routes to approach using the in-situ method. The particle method, where you add the carbon source and the modification together in, for example, silicate particles and glucose into a mixture. You can heat these and break these down to form glucose silicate aggregates, and then you add this to the growth media and ferment your bacteria, and this is in the aim to produce a modified cellulose with the aggregates and the nanoparticles incorporated into the cellulose fibers. And the chemical modification method, which was most recently done by Ming Gao, where you take a modified form of glucose, you can react it with the chosen modification that you want, and you use this as the carbon source to create the modified cellulose, and the aim is to incorporate the chemical modification directly into the cellulose chains. The process I am using to modify my bacterial cellulose is shown in this slide. So to begin with, we have the reaction to produce the modified glucose or the particle. Then we have the washing and cleaning of the products, just to remove any unreacted or aggregate that we don't want in the mixture. The inoculation fermentation of the cellulose, which usually takes around 7 days at 30 degrees centigrade. The pellicle formation, the washing of the pellicle, several different methods to try this, but we usually use the 1% sodium hydroxide method, boil it for 2 hours. Then we have pictures of the washed pellicle. As you can see, it's very different from the unwashed. And then finally, the drying step using either the oven method, which is usually 60 degrees overnight or until there is no more weight change in the cellulose pellicle, or the freeze drying method, which can be used to make a bacterial cellulose aerogel. For the hydrophobicity testing, using a droplet size analysis machine and measuring the time it takes for the water droplet to be absorbed into the material, and also the contact angle measurements, which are currently undergoing analysis. So currently we have the droplet absorbance times of a few of the modified cellulose. The TEOS is the tetraethyl orthosilicate, and the TMOS is the tetramethyl orthosilicate, and of course the plain bacterial cellulose. And as we can see, the tetraethyl orthosilicate from the 2325 strain has shown a marked decrease in the hydrophobicity, which isn't exactly intended, but it shows that this method of incorporating the modification has affected the properties of the bacterial cellulose. During the EDX analysis, we found that the tetraethyl orthosilicate from the previous one that showed the marked decrease in hydrophobicity also contains the largest amount of silicon. This is just a nice added piece of data to show that the silicon content has a direct effect on the water absorbance of the cellulose. These are some SEM images of the cellulose. So the acetic acid modified cellulose is very, very closely similar to the plain bacterial cellulose. As we can see, the tetraethyl orthosilicate has larger and much more prevalent amounts of silicon than the other silicate modifications. You can even see some particles that would be considered nanoparticles coating the surface in the top right images. And the tetramethyl orthosilicate also shows some nanoparticle aggregations on the fibers. You can see this is on the nanoscale, as each of those fibers is roughly 50 nanometers across, and some of the aggregates are even smaller than that. So in conclusion, the SEM analysis has suggested that there are silicates present within the modified cellulose structure, and the water droplet analysis did show that the hydrophobicity decreased with the incorporation of silica into the fibrous network, which wasn't intended, but it has shown that this method can be used to modify the properties of bacterial cellulose. And while some of the aggregates are not directly bound to the cellulose, they are directly incorporated into the fibrous network, even after several washing steps, which has shown that this method has changed this material. So for the future plans, we wish to trial other hydrophobic modifications, such as long-chain fatty acids attached to glucose. We also wish to use click chemistry to functionalize cellulose fibers with azide groups. And this will allow us to use the whole wealth of click chemistry enabled molecules and attach them to cellulose by just modifying one glucose with an azide, and the reduction of the modified cellulose to the individual fibers, which will allow us to craft cellulose materials with different morphologies and enable it to be ready to be used in methods such as electrospinning, which can give us whole new topologies and even surface changes, which can affect the hydrophobicity and many other properties. Thank you for your attention. If you want any of the references or just for any inquiries about my research, don't hesitate to contact me at this email. And thank you for your time.
So thank you, Chris. And now I'd like to welcome Paul James.
Well, thank you very much. And thanks very much to the organising committee for letting me talk to you today. The title of my talk is Project Perchlorate and Beyond. And I'm actually going to start with the Beyond and then go back to Project Perchlorate. So I'm just going to give you a very quick intro to synthetic biology and engineering biology, mainly just trying to introduce some of the things I'm going to talk about when I'm going through the work today. Then I'm going to talk about some work that we do in our lab to look at non-model organisms and bacteria that sort of survive and thrive in some weird and wonderful environments, and then get back to this Project Perchlorate that was actually a student project done a couple of years ago at the University of Exeter as part of the iGEM competition. So this is the sort of standard definition you'll see when people talk about synthetic biology, but it's the application of engineering principles to biology. And we're trying to create or design and build, I should say, not create, I get told off for create a lot of the time, new biological parts, devices and integrated systems. And what we're trying to use is this design, build, test and learn cycle. So you can see Up here, usually we're selecting some sort of bacteria, we're selecting some pathways of interest that have been found to do a particular job that we're interested in. And then it's about how we can go through sort of iterations of this cycle as quickly as possible to try and design and have a working system that can be used to, in the example I'll give later, to reduce perchlorate. And I suppose, what do we mean by applying engineering principles? Well, it's that design cycle that we're talking about, but we're trying to use a standardized library of parts. So we're trying to break each of these biological components down to the very minimum part, and be able to use those in a way that we can start to build up more complex systems. So in the same way, sort of electrical engineers will go to resistors and capacitors and know everything about that particular component and use that to build up a system, we want to be able to do that, but hopefully using biology. Modeling is a huge part of what we're trying to do. So obviously, in terms of time needed to do some of these things, modeling is a huge way of speeding up that design cycle. So can we go through some iterations of this before we even get to the biology and to the wet lab work? And then somebody once told me that synthetic biology is underpinned by three things: characterization, characterization, and characterization. And basically what we want to do is we want to be able to characterize these parts so that we know exactly how they're going to perform in a wide range of different environments. I think if you can go and have a look at some examples from Imperial, but they're trying to build up sort of standardized parts pages, much like the iGEM registry does, so we can look at have all this information available when we start to build up our models and look at these different things. And I once went to a really interesting talk from Chris Voigt in America, who talked about using these biological components to build up these logic gates, and can we build a cell that can get to the same sort of computing power as the Apollo 11 guidance computer, and that's one of the schematics there shown at the bottom. So what could synthetic or engineering biology do for space exploration? Well, this came from a very interesting paper in Grand Challenges in Space Synthetic Biology. And hopefully you can see that each one of these biology is able to do very easily. We find microorganisms that are able to survive and thrive in a wide range of different environments, utilizing substrates that are quite often man-made, but they've been in contact with for a while, and they've developed ways of coping with that. The development of a PET degrading enzyme from organisms that were found in PET degrading environments shows that their biology is able to adapt to these things, even if they're man-made, quite quickly. Obviously, biology can be used for manufacturing. So we have a wide range of different things now that are made by engineered cells. Hopefully, I'll show you a little bit more on the life support. But we have bacteria or microorganisms that are able to take CO2, make oxygen, make the sort of things that we're needed. Space medicine, I think there's been cell-free systems shown now that can create interesting chemicals that have medical applications. And I'll talk about cell-free synthetic biology in a little bit, and then things like space cybernetics and terraforming. So can we develop organisms that are able to sense their environment, they're able to respond to changes in environment, and what can we use those for in the end? So as I said before, I work mainly on non-model organisms. So typical organisms that we use in the lab, such as E. coli, or Bacillus subtilis, are great because we know a lot about their biology and we know a lot about how they perform within a lab setting. But a lot of times we find these very interesting bacteria that have been able to adapt to a particular environment or been able to produce a particularly difficult compound that chemistry takes a while to make. And what we want to do is be able to harness those different parts that the bacteria are able to do easily, and then maybe supplement them ourselves and try and bolt on bits that allow us to do that maybe quicker or in a more effective manner. So I sit within the building metabolism theme of the Hub for Biotechnology in the Built Environment. I have to remember that one. But so what we're trying to do there is we're trying to take waste and make a product of interest. So we're trying to take some of the things that we regard as waste and use them for high value products and things like that. So non-model organisms, they're highly adapted to extreme environments a lot of the time, and they're able to carry out very complex tasks. And what we're trying to do is build toolkits of parts. So those parts that I talked about before, we're trying to create new ones or borrow ones from well-characterized species and use those to be able to do a wide range of different things, not just waste degradation, but can we use them to produce biomaterials or engineered living materials that are able to respond to their environment. And there's a number of things that we're looking to do with that. So I also spoke about cell-free synthetic biology. So we're trying to move away from having to have that living cell and keep it living. And so what we do is we use wholesale lysates in my group. So we're not working with an engineered enzyme system. We're looking at using all the native machinery of the bacteria. We lyse that, so we take all the living components out, and then as part of that cell-free lysate, we have a lot of the machinery that's needed to produce proteins or produce pathways and things like that. And they're brilliant because they can be used a lot of time on the microfluidic scale. They can be dried down and they're very stable. We can use them for a wide range of different purposes. And you can see these now, they're starting to come online. Things like portable diagnostics and biomolecular manufacturing are just some of the areas that these cell-free systems are starting to be used in. And they could be particularly useful for some of those other areas that we're interested in. So back in 2018, we ran a competition. Well, we took part in a competition called iGEM every year. So we take an interdisciplinary team of 10 students, and they came up with a project. They designed it all by themselves. They implemented the lab all by themselves, and we just tried to help them with some guidance around what was and wasn't possible. So they were really interested in this idea of sending people to Mars and having people on Mars sort of within the 2030s. But what they wanted to do was take a problem that they'd seen locally and see if that could be applied. So we had some mining waste areas down in Cornwall that had high levels of perchlorate, and they were leaching into the water, and they were looking at trying to detoxify them. And as they were looking at this, they saw that Martian regolith actually contains 1% perchlorate, so quite a high proportion of perchlorate. So we know perchlorate is toxic to humans. It provides an extremely hazardous environment, especially when it can leach into the water system. And it's produced as a waste from a number of industrial processes, but some of those mining processes are particularly bad at producing these. But hopefully what you can see from that equation there is that if we are able to detoxify this, then we can produce molecular oxygen with it. So their aims, so we had 10 students for 10 weeks, but the students are nothing if not ambitious. So they had very lofty aims. So within those 10 weeks, they wanted to produce an E. coli that was able to reduce perchlorate to molecular oxygen. They wanted to be able to harvest oxygen from this process. And then as part of that, they also wanted to design a bioreactor that contained this reaction on Mars. So in terms of perchlorate reduction, we see quite a number of species of bacteria that are already able to do this. And they do this through this PCR ABCD system that's able to take the perchlorate and produce chlorite. And then they use a chlorite dismutase to then break that down into oxygen. So we know that system exists. We know, so we have already those parts that we can start to draw on. And there was a number of different bacteria that had these systems. They chose the PCR ABCD system from Azospira sulum. And then they chose 4 different chlorite dismutases. So from that sulum again, and then from, I think, 3 Dichloromonas species. So once again, you can start to see that we're able to harness these parts from other organisms and try and implement them within our bacterial system. So as I said before, we've got the PCR ABCD system that's able to produce these enzymes that are responsible for the reduction from perchlorate to chlorite. There's also a couple of sort of helper enzymes in there. So one of the enzymes needs a molybdenum cofactor. That's what one of these genes is responsible for. But we also need to be able to move this across membranes. So we need to move this from the cytoplasm into the periplasm. And we wanted to see if the native signal peptides, so we were taking those pieces of DNA as a whole from the different organisms, were those native signal peptides being able to be used within our E. coli system. So as I said before, what we're trying to do is we're trying to build up parts. So we're trying to use sort of established parts and trying to apply them within these different concepts and within these different engineering challenges. So these are the parts that we used. This is the synthetic biology output language. So these are the sort of standardized parts, libraries that we're trying to use, and standardized systems we're trying to use. I suppose what's important to recognize, I should do that on here for the people. So we have promoter, so something that initiates the production of the particular protein. And we have our protein of interest, or our open reading frame, and then we have our terminator, so something that says to the bacteria, okay, you can stop now, you've produced what we need you to produce. So the chloride dismutases were very easy to do. They were just a one enzyme system. We just put them in front of one promoter. The 4 enzyme system was a little bit more difficult. Obviously, with trying to get that within a plasmid and put it into the E. coli just becomes a little bit more challenging. So what we tried to do was just have it as one long piece of DNA with different ribosome binding sites. So this is different initiators as part of that promoter sequence to say, okay, yeah, I need you to make this piece of DNA for me now as well. So once they had the parts, they wanted to do some modeling. As I said, it helps to speed up that design cycle. And I think what they really wanted to know was, well, is this usable? Okay, we know we have people that are trying to breathe in oxygen. It has to be a robust system. It has to produce enough oxygen for however many people will be sitting within that area. And they worked out, I think, for sort of the standard habitat size, they would need a 48-litre bioreactor to produce enough oxygen for one person. So obviously there's some more challenges there. There's some improvement that needs to be done to that system to be able to make that usable, but it's good to have that information at the start. You know, there was a feasible working system that we could get, and that was with a working concentration of 1 millimolar per chlorate. So, you know, we're starting to build up, okay, well, how much do we need to start with, and then how much oxygen can we make as we move through? So, as I said before, we take perchlorate and we make chlorite. So chlorite's is even more toxic than perchlorate. So that's where that an even bigger issue lies, because once we've taken perchlorate and made chlorite, we need to get rid of that really quickly. So we need the chlorite dismutases that have the fastest rates of reaction. Otherwise, we're going to kill our bacteria quite quickly. And obviously, they wanted to see what sort of concentration of perchlorate they could get the bacteria to grow on, to try and look at, you know, was this feasible in a perchlorate-rich environment? So to start with, they just did some growth of E. coli on the different concentrations of perchlorate and chlorite. So as you can see, if we're working on a 1 millimolar system, then E. coli is quite easily able to grow on those different levels of perchlorate. Okay, it gets a little shaky when we get up to 200 millimolar, but we still see some growth of that E. coli. But as you can see, chlorite is a whole different beast. So once we get above 2 millimolar, then we start to see complete death of that E. coli system. So once again, we know there that okay, it's probably able to handle 1 millimolar, but not that well, and we need to get rid of that as quickly as possible. And obviously, what we don't want is a local buildup of that chlorite concentration, because that can cause cell death as well. So to start with, what they wanted to do was look at the expression of these chlorite dismutases. So this is just a gel on the side here that just shows the different levels of expression. So you can see that C, we have A, B, C, and D here. C is able to express really well, and it's also our most active enzyme. So we were able to see these enzymes take chlorite and produce molecular oxygen, which was really exciting for the team, and sort of showed a proof of concept of that system. So they then had to start looking at expression of this PCR ABCD system. So multi-enzyme expression is obviously a lot more challenging. There's a lot more biological information for the E. coli to have to sift through and work out and allocate resource and things like that. So what we did was tag each of these enzymes with just a different tag. It just allows us to see it within this Western blot system. And what we saw was expression of C and D, but none of A, which is weird because our promoter, as we said before, starts initiation here and then moves along. So PCRA should have been the first enzyme that was expressed. So just because we don't see it on this gel doesn't necessarily mean it wasn't expressed, but it might be that it's expressed with the tag hidden, or that what we saw was misfolding of this enzyme. So we have to do or the team, as further work would have gone to have a look and see how they could have improved this expression, or what about this PCRA system needed to be improved to kind of get it to work within this system. So the team weren't actually able to see expression of this system as a whole. So we never got to the point of taking perchlorate and making the chloride. But it's definitely something that they wanted to kind of move on to. But as I say, they only had a very limited amount of time, especially within the lab. So as I said before, we wanted to look at signal peptides. So signal peptides are exactly what they say on the tin. So they're a very small tag that the cell says, okay, I need to move this across membranes or around the cell. And what we did was take the native signal peptides, so the ones from not from E. coli, but from the different organisms that we took these parts from, and see if they were active in E. coli. And most of them were, so that was really good. It meant that we didn't have to do too much extra engineering to get them to go across that space into the periplasm. So that's as far as they got with the biological characterization. But one of an important element of this work, I think, is and especially as part of the iGEM competition, is trying to take projects that have got to a certain point further forward. And I think there's been 2 teams that have used some of these systems now within the iGEM competition to try and move this work on a bit further. So what they also wanted to do was look at bioreactor design. So as I said before, we had an interdisciplinary team. We had some engineers and some computer scientists that really wanted to get into the bioreactor design and look at how we can marry up some of the biology with some of the engineering that would be needed to deploy these things. And this was the sort of design cycle that they tried to go through. So, similar to the design cycle that we have for synthetic biology, they wanted to make sure they went through and tried to use the same processes. They weren't able to do too much building of this within the time frame, but they certainly wanted to try and get to a conceptual design that they could use. And they did this sort of in collaboration with some members of the European Space Agency, by talking to some people at NASA, and talking to people at Southwest Water, so our local water company, to kind of look at how they deal with these wastewater treatments and things like that. So trying to learn from experts in the field and then trying to apply it in a different situation. And they came up with these needs. So it needed to be able to contain the GM E. coli. Obviously, we don't want contamination coming in, but we also don't want any contamination going out. They needed it to be resource efficient. So obviously we know that resources are scarce, and we need to make them as resource efficient as possible. Obviously, it had to be robust. So, if we're producing molecular oxygen for people that are within that environment, then it breaking at any moment is a problem. They wanted it to be largely automated, so not too much interaction had to be done. And then they issued this 200 litre capacity, and that was based on those initial calculations of 48 litres per person. And they came up with and used a number of designs that sat within the literature. So they had sort of just bacteria that was just flowing in and around this system. And then, somebody pointed out that would easily flow out of whatever waste ports were coming out. And then they tried to use an immobilized bacteria setup. There's quite a bit of evidence that shows that applying small electrical currents can help some of these electron transfers and things like that. So they were trying to sort of marry all this together. And eventually came up with a bioreactor design that they thought was able to meet their needs. So this sat with the E. coli being trapped within an alginate bead that it was able to survive in, mixing with water and regolith, and then hopefully some oxygen coming out. So this was all conceptual. They weren't able to actually build any of these, but hopefully by having these discussions and this stakeholder engagement early on, then we can start to look at, you know, what's feasible and what's not feasible from a lot earlier. And I think we talked about that in the last session, which is really good. So just to went over what the team did. So yeah, they were able to show activity of 3 of the chlorite dismutases and able to use that to produce oxygen from chlorite. We saw partial expression of the PCR ABCD operon, but obviously that needs to be improved. And they were able to demonstrate that the native signal peptides could be used in E. coli. So the great thing about iGEM is that we just send the students off and they don't really care who they contact. So they were able to contact a lot of different people just by sending emails saying we're a team of students, we're looking into this, we're really trying to produce something that could be used. And we don't want to do sort of our research without trying to appreciate and trying to use some, if not all, of your knowledge. So they were able to contact a wide range of different stakeholders, and they were all very generous in providing their time, and most of them are up on that shot there. So I suppose I just wanted to show that again because I thought it was really, really interesting, and I think hopefully you can start to see how some synthetic biology and some of these non-model organisms and cell-free systems can start to contribute towards a lot of these different areas. So just in terms of thank you from me, I just wanted to thank everyone in the HBBE and in particular the leads on the different projects and my PhD student, Matt Rogan. So I didn't actually talk about any of his work as part of this, but he's working on trying to build some tools for one of these non-model organisms that we work with that's able to survive at very high temperatures. So if you have any questions, then that'd be great. If not, you can always contact me at the bottom.
Thank you so much. Well, so we have about 5. Oh, am I speaking to? Thank you very much, Paul, for this. We've got about 5 minutes for questions.
Perfect.
So you just stay there and hello. I think this one. So if we have any questions, please raise your hand.
Hi, thank you so much for the fascinating talk. I have like a zillion of questions. I have to choose like maybe 2. So one is clarification, actually. So you were talking about perchlorate turning into chlorate, right?
Yes, yeah. So
and you said it maybe even more toxic. So what's like the metabolic, like the ideal metabolic process? What are you hoping to achieve? Is there oxygen released?
Yes, yeah. So the idea would be that yeah, so it goes perchlorate to chlorite. That was the PCR ABCD system. And then the chlorite dismutase, that CLD enzyme, is able to take the chlorite and then make molecular oxygen. So I think what we're hoping is that that chlorite doesn't hang around too long. The chlorite dismutase is produced in large amounts. It's very active, so that hopefully that chlorite is immediately, sort of very quickly used to produce molecular oxygen, and then that could be released to the environment.
Great, thank you. So my other question, I chose one of 2, maybe the second one I'll ask you individually. So bioreactor is like a really charming idea, right, for like space construction, but it uses a lot of water, right, in case you put it. So what do you think? There were some ideas in previous talks about using like waste, maybe human waste as an input for bioreactor, maybe like additional metabolic processes before it can be used by bacteria. What do you think about that?
Yeah. So I suppose to link it back to the research that we're doing within the HBBE, a lot of that is trying to look at all waste streams. So that includes human waste. So we're hoping that we can modify some of those bacteria to be able to use all parts of those you know, of that waste stream. I know there's things such as microbial fuel cells that are able to produce electric currents from human waste, in particular urine, I think. So, you know, I think there's probably quite a bit of work that could be done to modify these bacteria, or have bioreactors with multiple bacteria within them that are able to sort of utilize all of that waste. So we can use the water element of urine to leach some of this perchlorate into, and then have that as a sort of a system which the E. coli could grow. And then also what we don't want to, I suppose, what we want to make sure is that we're getting everything useful out of that system. So we don't have, what we don't want is tanks of stuff sitting anywhere, because obviously that's no good to anyone. So how much of that system can we use? I think bacteria, well, I'm biased because I work on bacteria, but I think bacteria, you know, have been shown to survive in all of those environments. So I don't think there's any reason why we can't try and use some of these bioengineering techniques to make them at least produce something useful at the end, or interact with something that can have a useful output.
Great, thank you. I'll keep my other question.
Okay, yeah.
Thank you very much. Yeah, so it's very refreshing to see some synthetic biology here, because I feel that there are like a lot of fields converging, and synthetic biology, you know, personally, I really believe in synthetic biology, and it's great to see also like some iGEM contribution on this. So how are you continuing the work done at iGEM 2018?
So unfortunately, at the moment, that's not really continuing. So yeah, it was kind of at the time I was working in a lab that just worked on biofuels, but with the only stipulation that we gave to our iGEM teams is that they could never work on biofuels, because it was, well, yeah, for a number of different reasons, but IP contamination being one of them. But so what we wanted to do was sort of try and get some of the lab members to stay on and work on this, but funding wasn't particularly available at the time. But I think as hopefully now, as when we're looking at this building metabolism work, we can start to bring some of that in, because obviously, as I said before, this wasn't just a problem for people going to Mars. This is a huge problem for sort of areas surrounding industry, and you know, not just here, but all throughout the world. So I think that's definitely something that we'd like to do, is to go back and look some more at some of this.
So that would be the major space application of the James lab you are working on?
Well, yeah, I suppose the waste degradation hopefully would play a huge role in that. So yeah, so hopefully, so I work on a particular bacteria called Geobacillus that's able to grow on a load of different waste streams and able to survive in quite a few different extreme environments. So I think hopefully that could have applications within those bioreactor systems as well.
Thank you.
Thank you very much. Are there any more questions? Do we have questions online?
This question is from Gurhan. He asked, have you ever thought about the ethical part of modifying, killing living organism? He's also collaborating with them in his research lab, but he feels he's killing them. So what do you think about that?
Yeah. So I think, you know, a major area since the sort of conceptualization of synthetic biology has been responsible research and innovation. You know, we want to make sure that what we're doing sort of in a controlled manner, and we're doing it for the right reasons as well. So you know, I think we've worked with Carmen, and Carmen's worked with a wide range of different synthetic biology labs to look at, you know, what are we doing, why are we doing it, are we doing it in a responsible manner, and how we can sort of continue to do that as we move from synthetic biology to engineering biology. You know, I think as part of a lot of grant applications for synthetic biology, that is woven into it. You know, when you need to demonstrate that you are thinking about these things, and I think it's probably you know, one of the more sort of progressive emerging technologies when it comes to things like that.
Thank you very much. I'm sure there are more questions. I'm afraid we now have to, yeah, start our lunch program.
Our next speaker is Aled Roberts, who's a research fellow at Manchester University at United Kingdom. And he will present biopolymer inorganic hybrid materials for extraterrestrial habitat construction. Please welcome Aled.
Thanks very much for the introduction. Thanks for having me. My title has changed slightly from the one that's in the document. As you might be able to infer from how plain my presentation is, my background isn't art or design. My background is science. Specifically, I did an undergraduate degree in chemistry, and then I kind of pivoted towards materials engineering in my PhD. Decided I would then kind of immerse myself with a load of synthetic biologists, and I've kind of been a token material specialist in the area of synthetic biology since then. So yeah, the new title of my presentation is Regolith Biocomposites for Transplanetary Habitat Construction. So off-world habitats on the Moon and Mars, I look at relatively near-term space exploration. They'll need relatively thick walls and ceilings to protect the inhabitants from radiation shielding and also micrometeoroids. So the Earth is protected by a geomagnetic field, which blocks most radiation from the sun, from the galaxy, and obviously means we can inhabit the Earth without too much difficulty. It's a different story on the Moon and Mars because they don't have this global magnetic field. So the radiation exposure there, if you have sustained habitats, will cause problems. It's above safe limits. And if there's a solar flare and you're unprotected, it will kill all the crew. They'll have acute radiation poisoning and die. So we need, well, the most sensible solution really is to have relatively thick walls or ceilings, or have some other type of shielding material, maybe live underground or something. But that poses a problem because it's not feasible to transport bulk construction materials to space from Earth. So it costs about, depending on the launch system, about 4,000, currently about 4,000 to 80,000 pounds per brick just to low Earth orbit. And obviously if you had to go from low Earth orbit all the way to Mars and land your construction materials, it's just prohibitively expensive. So as we've heard in many of the talks over the course of this conference, we really need to utilize in-situ resources where possible. So this is known as in-situ resource utilization. And one thing both the Moon and Mars have of in great abundance is regolith, which is loose, unconsolidated dust and dirt. So if there was some technology to bind this together to stabilize it into a solid construction material like a brick, a concrete, it could serve as a radiation shielding material, but also a construction material. So there's quite a few proposed technology options to achieve this, listed them here. So for example, you could heat the regolith up until it forms like a lava and then cast it into like blocks. You could potentially produce a type of cement on Mars and then make like a concrete. We've heard the potential to make synthetic polymers, maybe from atmospheric CO2, or there's other binder options like sulfur obtained from regolith or ice deposits. But these technologies all have like some significant drawbacks. So for example, heating the regolith into a lava and casting into a block will obviously use like huge energy, and that's a problem on the Moon and Mars because if you need a lot of energy, you're going to have to like take additional solar panels or whatever system you're using to generate power. You'll need more of it, and that will add to launch mass, add to the cost, and kind of offset the benefits of utilizing in situ resources in the first place. So so I guess like because you're all here, you all kind of already kind of agree that biological systems are a potential panacea. They potentially solve this problem, and one of the big advantages that biology operates under ambient conditions, you know, like conditions compatible with life by definition. And if we are going to send humans to the Moon and Mars, we're obviously going to have all the kind of life support systems we'll need to sustain biological systems because humans are going to be there. So it, you know, having producing things using biological systems seems to be quite a neat solution. And the way I see it, I think I can really see flexible bioreactors being like a really important component on any kind of sustained kind of habitation of like the Moon and Mars. So yeah, we've heard loads of presentations over the course of a couple of days, and I think like flexible systems, maybe taking some like frozen stocks of different host organisms. I think some kind of photosynthetic organism will probably be the most feasible. So we've heard about the Melissa project, where you're growing algae as like a significant kind of food source for the astronauts. But if that was engineered, we heard in a presentation before, it could be used to make rocket fuel to power your Mars ascent vehicle back to Earth, produce oxygen, deal with the perchlorate issue, as Paul mentioned yesterday, and also, generate just all sorts of pharmaceuticals and chemicals just through engineering the organisms to produce enzymes and convert resources you can obtain on Mars, water, carbon dioxide, nitrogen, fixed nitrogen from the atmosphere, into useful chemicals and materials. So for example, say if your astronaut gets some kind of illness and you've not taken that specific medicine to treat that illness, scientists on the Earth can work really hard to come up with how to engineer one of your bacteria to produce that pharmaceutical, and then you can produce that, and would not then need to like transport it, so it could save on launch mass. So yeah, the question is, can we utilize these living organisms for transplanted construction? And I guess that question has already been answered, and if you're in this conference, and we've heard this time and again over the last couple of days. So maybe a better question is, what system will we ultimately end up using? Because there's lots of like potential methods we could apply, but which ones, will we ultimately end up like applying at a significant scale? So my specific for these things called Regolith biocomposites is quite a simple system compared to like any kind of like living material. Essentially, at its core, I don't know if this. Oh, never mind. At its core, essentially, we're utilizing like the natural adhesive properties of proteins and carbohydrates. So proteins and carbohydrates are sticky, they stick stuff together, and if you combine it with an inorganic aggregate like regolith, you can make a regolith biocomposite. And our initial focus was to engineer a host organism to produce spider silk proteins, and then these would be recombinantly expressed, purified, concentrated, and the idea was, we could have a bioreactor pumping out this very sticky protein, which we could then combine with regolith and make into like bricks, and obviously that bioreactor could have other functions as well. But then we found, just through a control experiment, that bovine serum albumin, which is very common, very cheap protein, relatively cheap protein, which is used for all sorts of like control experiments, actually did better. So we're really like confused about this. But then we did a literature search and found that some other people had already investigated this. They literally used bovine serum albumin with regolith to make a biocomposite material, and it was surprisingly strong. But they basically came at it from like an engineering materials perspective, and they were just like, oh, we just picked this random protein because it was cheap off the shop. But when you think about it, so this bovine serum albumin, it's obtained from cow blood, and it's not feasible to take cows into space. It's going to be a nightmare if they're floating about, and getting them to use the toilet system is just going to be a nightmare. But humans are also animals, right? And humans are going to be present by definition on any kind of crewed space mission. So could we like use the human equivalent of this protein, human serum albumin? So we kind of explored this kind of concept, considering the humans on the planet as an in-situ resource. And yeah, we looked into it, did some thought experiments, and then we actually went forward with the real experiments. And yeah, it's quite interesting because a human body produces about 12 to 25 grams of human serum albumin per day. So we're actually quite an efficient bioreactor. Your body's constantly making this protein and breaking it down, and its function in the blood is just an osmotic regulator is its main function. So it's very cheap protein that your body's just pumping out. So you can harvest it through blood plasma extraction, put the blood cells back in the body, and utilize that potentially as a binder for regolith. So I won't go too much into the science, but this is kind of a schematic showing what we did. Very simple system. We got this human serum albumin, mixed it until it dissolved at a high concentration, packed some syringes with regolith, and then just infused the resin in, which was just the concentrated solution of this albumin, and then dried it, and then it formed these biocomposite materials, and the compressive strengths were remarkably high. We did a bit of, so 40 megapascals for comparison is about twice as strong as an ordinary brick. And we investigated the mechanism. We found that it was driven by hydrogen bonding interactions, which makes sense. And there was no other kind of feasible bonding mechanism that would be occurring as far as we know, because we didn't add any covalent cross-linking agents, we didn't add any ionic bridging agents. And then when we investigated and found it was due to hydrogen bonding, we added urea, which is a substance, it's the main solid component in urine. And we thought this would be great to test our hypothesis, because urea forms lots of hydrogen bonds and it affects the folding and unfolding of proteins. And we thought, if our hypothesis is correct, urea will have an effect on these materials. And we thought it'll probably make them weaker because it'll be disturbing, the whatever's going on, but actually made it stronger. So that's how we got to the 40 megapascals figure. Adding urea made the material stronger. And obviously urine is going to be a byproduct, a waste product on any kind of long-term trip. We're making huge amounts of it. Most of it's just going to go back into the Melissa style, it's going to feed the plants again. Where if we can utilize it to make, construction materials, all the better. So yeah, human serum alum could be used as a binder for regolith, but maybe not should, because it's going to have drawbacks. It's going to affect like the health of the astronauts. But this is just a scheme kind of showing the concept. So you can obtain nitrogen and carbon dioxide from the atmosphere of Mars, water from regolith, use that to grow food and oxygen through whatever system you're using. That food feeds the astronauts, and then you can tap off surplus human serum albumin and like their urea to potentially make these biocomposite materials. And yeah, it overcomes many of the issues faced by other technology options. So it's not a low and not high energy process. You don't need to bring significant additional binder production equipment. Like you don't have to produce like polymers from the atmosphere or anything. The humans and all the support systems are going to be there for them anyway. But yeah, like I said, the main drawback is it's harmful for human health, and for that reason, it almost certainly would never be actually employed. But you could maybe imagine like an Apollo 13 type situation where there's an emergency and you need to just MacGyver up some concrete real quick, and just knowing that blood and urine could potentially make like a material with a high compressive strength could be useful in certain situations. So yeah, knowledge never harms anyone. But then we thought, well, what else can we use? And it turns out blood was historically used as a binder. This is called bois d'urcy. It's like French. I'm not sure if I pronounced that right, but basically it's cow blood mixed with sawdust and compressed. It made like a plasticky material. And we did like a historical kind of like literature survey to see if blood works and what else can work. Is anything else that's more feasible? And we found, egg white was used as a binder, was used to like stick gold leaf onto materials, which lasts for like hundreds of years. But yeah, we thought chickens in space is also going to be quite problematic, probably less problematic than cows, but still probably not really that feasible. Collagen from connective tissue and skin was historically used as a glue. If you've ever heard the term sending the horse to the glue factory, that's where it's from. Obviously, we can't harvest the skin of astronauts. That would be horrific. But people shed skin flakes all the time. It becomes dust, and that dust is going to obviously accumulate in a closed system in like the vents and stuff. So you might have a reasonable amount of skin flakes from the astronauts, and what you're going to do with it. Okay, you can show it back to the food production system, but if you can use that as a binder to make a material, but we thought this is probably going to have the same issue in terms of like not that feasible in terms of amount we can make. And then we thought cheese. So cheese was used as like a glue up until World War II for like sticking wooden panels of aircraft together, so it makes a strong glue. Obviously humans are mammals, so we could potentially make human cheese, but it's definitely not an avenue I wanted to go down, so that's just staying in the thought experiment pile. But then the really interesting ones were gluten and starch. So the word glue and gluten have a common origin. It was used historically by the Egyptians, the Romans. It's like wheat paste, you use it for like paper mache and stuff, and the same with starch. Yeah, interestingly, starch can make a very, very strong glue. Aboriginal tribes still use it to stick feathers to arrowheads, and obviously quite a strong glue to stop the feathers coming off your arrowhead. So we explored this, and we thought starch would be more interesting because it doesn't, being a carbohydrate, it doesn't have nitrogen, whereas gluten is protein based and has nitrogen, so it's going to be better in terms of it's not going to be depleting your nitrogen. So this is just a schematic showing the fairly simple process. You get your inorganic aggregate, your regolith, mix it with starch powder, and add some water, heat it up, and this gelatinizes the starch, so it thickens it. If you've ever made like gravy, that's basically what's going on. Then we found the best way of doing it is to partially dehydrate it, compress it, and then fully dehydrate it, and then you're left with, we call it starcrete, so starch concrete, which is essentially a starch-based biocomposite material. And then we did some optimization through design of experiments, which is essentially like statistically augmented experimental design. So it's basically quite an efficient way to design experiments and do some optimization. And doing this method, we were able to get compressive strength as high as 70 megapascals. So this is like super high. It's within the domain of high strength concrete. So a really nice, strong material after that optimization. So yeah, just in summary, these regolith biocomposites are quite a promising technology for transplanetary construction. Starch seems to be a lot more feasible than human blood, and of the other systems we've analyzed, it seems to be definitely up there in terms of feasibility. And this is just comparing like the two, the astronautics, that is the blood one. We literally cut out the middleman and go straight from the Martian environment to produce food in the regolith to make this construction material. So it's quite a simple, elegant solution to the issue. And just one final thing, yes, I'm really interested in like art science, the interface of art and science, and we've got this ongoing collaboration. And actually, a lot of this work is kind of inspired and done with artists. And in fact, the whole concept of using starch as a binder was from a conversation with artists. They were just like, we were talking about the glues, and they were like, oh, rabbit hide glue was also used as, you know, stuff I didn't know. And they were like, oh, have you tried like starch glues? And I was like, didn't even know about that. And they handed me some like rice glue, and I tried it, and it worked. So yeah, that's going on. I'm really interested in this space. So thanks very much for listening. I'd like to thank all these people, in particular Anthony Hall, who I think I've left off that list, but so I'm thanking him personally. Happy to take any questions if I've got time.
Thank you so much, Aled. That was fascinating. Are there any questions on the audience? Yeah.
It's a good thing your research wasn't around when they filmed The Martian, to be honest. But I was just wondering, did you try different size of aggregates? See if that, because obviously the binder you use are quite liquid. So I'm thinking if the aggregate size are quite big, you might lose the compressive strengths.
Yeah, that's a really good point. So on the Martian thing, actually, in the book, when the guy gets stabbed, this is like the film The Martian. I don't want to give too many spoilers, but when the guy kind of has his accident, actually his blood, it's described in the book, like mixes with the Martian dust and kind of forms a seal. So actually there's kind of like a bit of the science in there, which is really interesting. And obviously he grows like potatoes as well. So there's kind of the connection with the starch. To answer your real question, oh, sorry. Oh, I've broken something, sorry. Hopefully everyone at home can still hear me. To answer your real question, yeah, so the aggregate particle size and particle size distribution is really important for any like of concrete. Generally, yeah, there's a lot of optimization in terms of like there's generally like two types of aggregate: coarse aggregate, fine aggregate, and the particle size distribution. Even the shape is very important. From my understanding, lunar and Martian regolith, because it's very sharp and jagged, it actually makes quite a good aggregate material. And the simulants we use actually do perform quite well compared to, you know, ordinary sand and stuff. But we didn't do any optimization. We thought, well, our justification, basically because I was too lazy for doing this type of experiment, was we want to keep the system as simple as possible. We don't introduce additional equipment or processes. So if we could just use plain old regolith and see how strong that is. Yeah, you're absolutely right. Like if we did filter the particle sizes and then recombine it in like a specific way, we probably could get even higher, better performance materials. Thanks. I appear to have broken the system, the computer.
You're the last one, so that's good.
Okay, phew.
Actually, do have a question in the meantime. Don't know if someone else knows a bit more about tech. I was wondering, you kind of gloss over the health issues.
how bad are they if we're thinking as a potential emergency? And also, is there anything else that would act as a binder? And do we have any of these, I don't know, chemicals or that we don't need that much but produce a lot of? I've never thought of it that way. So.
Yeah, so humans do produce other sticky substances which could potentially be used, but I definitely didn't want to go down that route either. In terms of the health effects, if you're doing it on Earth, it's not too bad. So basically there's plasma donation procedures and you can give quite a lot of it. I think you can, WHO recommends, I think up to 2 donations per week of like a couple of liters or something per go. So you can actually give quite a lot safely on Earth. The problem is in space, there's lots of health issues in microgravity. And I think a lot of it is related to the circulatory system. So if you've got like health problems just from being in microgravity for like 3 months as you're going to Mars, probably the last thing you want to do is to be giving your blood away. And we still don't know the effects of hypergravity, like low gravity on the Moon and Mars for extended periods. So it would probably not be good to kind of depend on this technique. Yeah, bloodletting is probably not that feasible really.
Thank you. So when I saw all the arrows going in one direction, instantly it triggered in me like, you know, like that was not a closed system or some kind of system that recycles the different atoms, molecules in the system. And yeah, it was just an observation that it might be like at some point maybe we are going to be trying to get the waste. So maybe there's going to be a competition for the waste, because I think that some research groups are focusing also like in urea, using it as a fertilizer for growing vegetables, or like also like binders, or maybe other groups are working also for to take that waste for other construction. So I mean, maybe in the future we are going to be competing for waste.
Yeah, no, you make a really good point. So in the paper, we have a more complete, more complex kind of systematic diagram of how we think it might work, and also how it would fit in with like a bioreactor. So yeah, it is an open system in that we will need like to take resources in from the Martian atmosphere, hopefully all from the Martian atmosphere and regolith. Because yeah, we'll be losing, for example, nitrogen if we're using like a protein as a binder. So we'll need to like fix that somehow. Yeah, the Martian atmosphere has like a few % nitrogen. It's not much, and obviously the Martian atmosphere is a much lower pressure than Earth. There's plenty of carbon dioxide, and obviously there's some water like just locked up within the protein, which you would have to kind of like regenerate. But yeah, as like Angelou's presentation was showing before, you need to kind of do a full kind of like mass balance analysis of your inputs, your outputs. And if this is going to be a significant output, you need to like balance that somehow. And if you're using urea for this and you can't use it for something else, luckily humans do produce a lot of urea. Like every day we make like lots of urine. So unless we're using it like for a really intensive process, there's plenty, and it's more of like an additive in this system. So we're not using huge quantities. The limiting step there would be the protein from blood. Yeah, good question, thanks.
I have one more question. I wanted to go back to perchlorates. Any of these ideas of making biocomposites from regolith, does it work despite the toxins, or you still have to solve the issue with toxin first before we can get to making biocomposites?
Yeah, that's a really good question. So I've not tested it. So the Martian and lunar regolith simulant I get, yeah, so the Martian one I think has the perchlorate issue, and the regolith simulant I get by default comes without perchlorates just because it's toxic to work with. But you can request them to add the necessary amount of perchlorate. So yeah, that's definitely something that's worth testing. If we were going to actually apply any of these, we'd have to obviously use perchlorates just to confirm it doesn't degrade the binders. And if it did, then yeah, we'd have to like process the regolith beforehand, like Paul's work was suggesting. So hopefully it won't be an issue, but it's one of the unknowns that would need to be investigated. Thanks.
Thank you so much. Are there any other questions? I think we have some time because we have to solve the screen. Yeah, there one more.
Promise I'll stall. At the start of your presentation, you mentioned radiation. I think this is more of a speculative comment rather than a question, but I think what your work kind of opens up is that we can potentially mix states, but also potentially mix new protein source. I'm thinking like maybe it'd be interesting to add like melanin proteins to see if that increases radiation shielding effects of the soil.
Yeah, absolutely. And I think like if you did go down the bioreactor route and made like engineered proteins, then yeah, there's definitely a lot of scope to also engineer the bacteria or the organisms to produce melanin to enhance the radiation shielding capability. Yeah, on radiation shielding, we didn't do any tests ourselves, but our kind of perspective was if you're making a material out of, you know, light elements or elements that are present in the human body, then that is probably going to help with the radiation shielding, because anything that's going to potentially damage your DNA or damage your body, if you've got all those elements as part of your shielding material, then hopefully it will get absorbed. And I think neutron radiation in particular, you need quite light elements to reduce the harmful effect of neutrons. So having relatively light elements is like your binder, carbon, nitrogen, hydrogen, oxygen, is definitely a good thing in terms of radiation shielding. Thanks.
Thank you so much, Aled.